All right, are there any questions? Okay, so I was going through this explanatory material, all right, last time, and I think we had a little bit left. Yeah, so we went through this rank sum test, all right, and I discussed that. Then I talked about the Chebyshev inequality, then hypothesis testing, right? And then the only thing that, there are only two things remaining, right? One is the derivation of equation 7, right? That you can see here. And I will show you equation 6 and 7, okay? So this part will fill in the gaps, okay, when we start looking at the paper, right? Going from equation 6 to equation 7. And then I already talked about the maximum likelihood estimation, right? And you have to calculate from the probability density function. You have to calculate the maximum likelihood estimate of the parameter C, and that's just done by setting the derivative of the log likelihood function to 0 and then solving for the estimate that is going to maximize the likelihood function, right? So any questions so far? So the only thing that was remaining was to show how the density function of the random variable y equals ax, where a is greater than 0 as a scalar multiple, is related to the density function of x, right? And that is actually an exercise in, in the first course in probability, right? And I'll just repeat it here for those who may have forgotten. So if you have a random variable ax with a greater than 0, you're trying to calculate the distribution function. So probability that the random variable y is less than or equal to little y is the probability that a times x is less than or equal to, this should have been little y, okay, which is the same as probability of x less than or equal to y over a. So you get the distribution function of x evaluated at y over a, right? So then you differentiate this with respect to y, right, little y, and then you get this expression that the probability density function for y is related to the probability density function for x, right, through this equation, right? And the way it's going to be applied is that if one of the two channels, the red or the green, has got a higher intensity, right, then instead of trying to do, test if the ratio is close to 1, you will test that the ratio is close to m. So recall that TK, which is the ratio of section 4 of the paper, and we'll get to the paper in a few minutes, is defined as the ratio of the red to the green. However, if the means have a constant scaling, right, then the new TK that we will be working with, say TK prime, is really m times this ratio, right? And for that, the density function would have to be changed using this relationship, okay? And that's going to be done in the paper. That's equation 11 in the paper, right? So in this case, you would have to estimate both m, right, the scaling factor, as well as the constant c. All right, any questions? So if there are no questions, I will now start discussing the paper, the actual paper. Okay, so this is the paper that we are interested in discussing. What is... Okay, no, it's better. Can you still read? You can read, right? You can read. Oh, you cannot read anything. Okay. Why is that? I mean, like, you can read now or what? Oh, you don't know what? I didn't understand.
No, I mean I can read it. I don't know whether the student did that.
But why is that? I mean, can you guys read the people at the end? You can? Okay, all right. Okay, so this is the paper that we are discussing, okay, ratio-based decisions and the quantitative analysis of cDNA microarray images, all right? So you should have no trouble reading the introduction, right? The molecular biology background and the cDNA microarray technology, right? This is stuff we have covered here, right, in this course. So you should be able to read through the first 2 sections of this paper just like that, there should be no problem, right? So this is all the biology. It's talking about complementary DNAs, hybridization, and so on. Okay.
So, is there any like more advanced technology than microarray?
Yeah.
Now?
Yeah. Right.
I saw the year
97. Yeah, it is 97. Yeah. So since then there have been improvements, and now you have that next generation sequencing. Okay. Because they can actually, what they will do right now, that is called RNA sequencing, right? RNA-seq, you may have heard of that. Yeah. Right, RNA-seq, so instead of doing microarray, they will just take the RNA and they will sequence it. Okay. And that's what Dr. Johnson, Charlie Johnson, right, does at the center. He has machines to do that. In fact, 2 or 3 machines. So now to analyze gene expression, they will not do this microarray. Okay. Instead, they will just go ahead and sequence it, and they have chopped up the RNA into many pieces, and well, actually from the RNA they'll get the complementary DNA. They'll chop it up, right, into many pieces and do this next generation sequencing, right. And so the number of times like each gene occurs, okay, or let's say a piece occurs, that is proportional to the concentration of the RNA. That's why it's called RNA-seq, okay, because they're going to sequence the RNA. But there you're not going to measure the level, the quantity of RNA, like you're measuring in the microarray using intensity, light intensity. No. Over there, it's the number of copies, okay, when you sequence it, because you're cutting it into pieces, right? So the number of copies is going to be counted. So if some RNA is present at a higher level, right, there'll be more copies of that, They call that those reads, okay? There might be 1 million reads or something like that, and so that is proportional to gene expression, right? So that is a more advanced technology to do this. Yeah. All right. So I'm not. Okay. Let's see. Let me put. Yeah. Let me make it a little bit bigger. Now it should be better, right? Or is the quality of this thing poor, right? So yeah, it is a 1997 paper, and it's talking about the recent development. So it's not recent anymore, okay? Because this is what 2014, okay? So it's not recent anymore, okay? For sure, right? So you can take a look at the abstract. It says gene expression can be quantitatively analyzed by hybridizing floor-tagged mRNA to targets on a cDNA microarray. Comparison of gene expression levels arising from co-hybridized samples, right, is achieved by taking ratios of average expression levels for individual genes. A novel method of image segmentation is provided, and this I will discuss, all right, to identify cDNA target sites. And a hypothesis test and confidence interval is developed to quantify the significance of observed differences in expression ratios. In particular, the probability density of the ratio and the maximum likelihood estimator for the distribution are derived, and an iterative procedure for signal calibration is developed. Okay. So again, these terms should not be new. cDNA is complementary DNA, microarray, gene expression, image segmentation we'll talk about, Mann-Whitney target detection, that's the rank sum test. Okay. A ratio density, ratio confidence interval, right? So this is just a brief introduction talking about the Human Genome Project and so on. Then there's biological background in cDNA microarray technology, right? And you should have no problem whatsoever, reading through this, because we've covered all this material in the class. So now, and here there is some justification for making that assumption about the constant coefficient of variation. Okay, later on they'll give some biological justification. And this is the picture, that's the slide preparation, right? So first you prepare this slide. Now you have these 2 different samples, okay? You do reverse transcription. From the RNA, you'll go to the complementary DNA, put in the different colors, let it hybridize, right? And then wash away the ones which didn't hybridize. And then you shine the light from the laser, right? And you get your image, and you basically go and do image analysis, right? And find out whether red is higher than green at each and every spot, right? So here, towards the end, right, they say, right, so towards the end, they'll say that in this composite image, one can see the differential expression of genes in the 2 cell lines, right? So you here for this paper they use two different cell lines. One is cancerous cell line, the other one is its precursor, which is not cancerous, right? Which doesn't cause tumors, and they're trying to see which genes are differentially expressed, right? So intense red fluorescent at a spot indicates a high level of expression of that gene in the non-tumorigenic cell line, with little expression of the same gene in the tumorigenic parent, right? Conversely, intense green fluorescent at a spot indicates high expression of that gene in the tumorigenic line with little expression in the non-tumorigenic daughter line. And when both the lines express a gene at similar levels, right, both the samples, then you will see the array spot is going to be yellow, right? Red and green combined, that's going to give you yellow. Now, visual inspection of such results is sufficient to find genes where there is a very large differential rate of expression, right? A more thorough study of the changes in expression requires the ability to discern more subtle changes in expression level and to determine whether observed differences are the result of random variation or whether they are likely to be meaningful changes, all right? So this is the rank sum test, okay? The next section is about image processing, because you have to identify you have the background, right? You have to identify the target, right? Because that's the place, I mean, you want to actually capture signal, because corresponding to hybridization, all right? Because you're really trying to measure the quantity of RNA, right? So the difficult image processing task right here is basically to look at the spots, right? See, because there is some pattern on this array, because there's a robot that has gone and put those spots, right? But it's not exact, right? So your image processing task is basically you want to find out at every spot, right? At every spot you want to be able to identify the signal, right? So how do you do that? Okay. So the way you do that is that, like, let's say you have a target mask over here, right? That is already known from before, okay, what the size is going to be. So outside that, it is all background, okay? Now, say if I'm going to identify the patch, okay, where the signal is there, right? The intensity over there should be significantly higher than the background, right? So how do I, find that out, okay? How do I determine which are the spots in this that I should pick up, right? So what you can do is you can, pick some spots in this background, right? Let's say 8 of them, right? 8 is the number that they used in the paper. So you pick those spots in the background, and then inside, like, you have signal intensity at all these different pixels, okay? You take the 8 pixels with the lowest intensity, okay? And now you do a rank sum test, okay? And your hypothesis is that, the two means are the same, right? Now, if it turns out that the null hypothesis about the means being the same, right, is not violated, right, then you have to look some more, okay? Then that means they really are the same. They're not significantly different, right? You have to look some more. If it is violated, right, if the null hypothesis is violated, that means with these like these eight lowest expression, intensity levels here, you get that the significant the intensity here is significantly higher than the intensity over there. What you're going to do is take the lowest among those 8, okay? Use that as your threshold. Anything above that is signal, okay? Now, if the test, the hypothesis, the null hypothesis still holds, it's not violated, okay? What are you going to do? Then you just throw away the lowest sample, okay? The lowest sample inside in here, okay? The lowest intensity level, throw that away. Try with the next 8 lowest ones, okay? And then keep on doing it iteratively, right? Until the null hypothesis is violated, okay? So that way you will pick up something that is significantly different from this background, okay? And what test are you going to use, all right? The test you're going to use is the rank sum test, okay? So you have the rank of all the signals that you're taking in here, the 8 of them, the rank, take that, then look at the tables at the back of the statistics book, all right? They have tables, all right? And you determine a level of significance alpha, right? How much error you're willing to make, right? Why do you always smile when I say about the tables, okay? I'm not meaning that you have to go and copy, okay? I'm not looking at them like Laplace transform tables, right? These tables are there because we have to use them, right? I mean, so you go ahead and so again, to repeat, the procedure will be as follows, all right? You'll pick some, you'll pick the, say, let's say 8 lowest intensities here, right? See if they are significantly higher than the background, right? Using the rank sum test. If not, take out the lowest intensity. Take the next 8 lowest. Keep doing that. It may so happen you keep on doing that and still you're not getting, the null hypothesis to fail, right? In that case, it means that there is no appreciable signal at this location, right? Because if it's the same as the background, you're not getting a significant difference with the background, you should not even consider that spot. Yes, sir.
How do you choose those 8?
No, you mean the ones here or the ones outside?
Inside.
Inside, you'll choose the lowest 8. See, there is intensity corresponding to each pixel. Okay, choose the lowest 8, because what you're going to do is once you found out, let's say this lowest 8, if it's significantly different from the background, right? You will take the lowest among them, the minimum among them, use that to do the thresholding. Anything above that is going to be signal, right?
And how you define the target mask?
Target mask is already defined. That's based on the shape of the thing. It says that in the paper, okay? It's based on prior considerations, maybe where the robot has put the stuff and all that. You have some idea. Let me just show it to you, okay? Okay. Are you talking about the target mask or the target patch?
Target mask, the red circle.
No. Let me see.
I mean, the region that you choose the 8
Yeah.
intensity.
Right. So anything above that, okay? Anything above that will be in the target mask.
Because I think the radius,
Okay. Yeah. Let's look at this, okay? So the difficult image processing task, all right, is to identify the target site within the target patch, okay? The target patch is that square, okay? So each target site is somewhat annular owing to how the robot finger places the CDNA on the slide and how the slide is treated. However, there is variability in this placement within the patch from image to image and from target to target. This variability can be so great that the target region is simply a collection of sub-regions, right, within the nominal circular target region, right? This instability in the target region is manifested in the irregular way the mRNA is hybridized to the target and the consequent irregular brightness pattern created within the target site, right? It is important that mRNA intensity be measured over these floor regions because only they correspond to probe hybridized to target, right? Because you are interested only in capturing the RNA, okay, the information that is coming from the RNA, right? Now, see, to overcome these difficulties, right, and he's giving reasons why standard morphological methods will fail, or conventional adaptive thresholding also will not work. So to overcome these difficulties, we propose a pixel selection method based on the Mann-Whitney test, and that is the rank sum test, right? So there are 3 key points associated with this approach. It associates a confidence level with every intensity measurement based on the significance level of the test, and if desired, it enables multiple readouts at different confidence levels. It meets the real-time requirement of the system, and it is distribution-free. We already discussed all this last time, okay? So you have x1 through xn and y1 through ym are independent samples arising from 2 random variables. Then you use the rank sum statistic, right? And you're testing for the hypothesis that the 2 have the same means, right? So a target site is segmented from the target patch according to the following procedure, right? A predefined target mask, okay, see it's predefined, okay, it's defined from before, is used to identify a portion of the target patch that contains the target site. So this is given to you from before, right? That's what I was saying before too. The target mask is based on the geometry of the potential target area and can be constructed from specially tagged targets or other strong targets. Example, the target mask is obtained by finding all strong targets, aligning them together, right? Averaging and then thresholding, right? So you'll use some other method. There's not much statistics involved in it, okay? So then after that, you randomly pick 8 sample pixels from the known background, right? Outside the target mask, call them y1 through y8, and select the lowest 8 samples from within the target mask as x1 through x8. Now you apply the rank sum statistic, right? W is going to be calculated, and for a given significance level alpha, you're going to compare it with the value that you get from the tables, okay? And you're going to laugh again. So anyway, so we choose 8 samples here for both foreground and background. The two numbers can be different. If you remember in the example last time, right? X and you didn't have to have the same number of x's and y's, okay? No, it could be m and n, right? It'll still work. But here he's taking m equal to n equal to 8, and the reason is because then the distribution is almost normal. Now, if the null hypothesis is not rejected, then we discard some predetermined number, perhaps only one, of the 8 samples, right, from the potential target region, and select the lowest 8 remaining samples from the region, right, and then repeat, right. And the Mann-Whitney test is repeated until the null hypothesis is rejected. When the null hypothesis is rejected, the target site is taken to be the 8 pixels causing the rejection, right, together with all the pixels in the target mask whose values are greater than or equal to the minimum value of the 8. Right? That's what I said. That's how you do the thresholding. Right? And the resulting site is said to be a target site of significance level alpha. Now, if it so happens that you keep on trying and the null hypothesis is never rejected, then it is concluded that there is no appreciable probe at the target site. Okay, you really don't have much signal over there. Okay, so furthermore, you can also put the requirement that when you're using the rank sum test, the target site could contain a minimum number of pixels for the target site to be considered valid. Okay. And it can be measured for flow intensity. So, you know, 2 examples are given for different levels of confidence, all right, on the next page, you know, the 2 pictures that are given. Or was it on the previous page? Previous page, yeah. So you can, see these are the targets that would be detected at different levels of significance. If alpha is 0.0001, okay, then you can see that you're not picking up some of these, like this one is not getting picked up, okay, this one is gone, okay. Whereas if you're going to allow for a larger error, right, that is the level of significance, then I mean some of these, like this thing is going to appear here, which was not picked up before, all right. All right, any questions? Yeah. Yes.
So, for example, the macro array is the size of 4,000 by 4,000, so we have to do like 4,000 times. No, 4,000 by 4,000 times.
For what?
I mean, the number of times that we need to do this processing is like 4,000 multiplied by 4,000.
Wait. Yeah, I would think you will have to do it, you know, why? Because at every spot, okay, it'll be a different gene, okay? So the intensity relative to the background could be different, right? So probably you will have to do it at every spot, yeah. And the background could also vary across this, yeah. And again, I mean, I haven't done this, so I don't know, okay, but I would think they would have to do it everywhere, because unless they make some assumptions about, even that wouldn't work, because, you know, sometimes the robot might, you know, move from one place to another, and there might be irregularity and all that, yeah. Yeah, you'll have to do it for all the different genes. There used to be 8,000, I don't know exactly how they did it in that paper, okay. Because this part, remember, this part they're not presenting as the main result, okay. This is like some image processing stuff, okay. So this is just an algorithm they're telling you to do, you know. Now, are you going to choose the threshold based on, you know, just one spot and apply it to all the other spots? Who knows? Maybe they did it, okay, if it was too much of work, correct? But in theory, you'd have to do it separately for each spot, you know, especially if the intensities vary a lot, right? Okay, any other questions? So that's how you basically did the image processing to get your target, right? And now you have to go and get the ratio, right? Okay, so you finished the Mann-Whitney test or the rank sum test, and now you have to get the probability density function of the ratio parameters, all right? So you now wish to use the gene expression ratio to determine whether gene expression differs significantly for the red and green samples, right? Such an approach is intuitive because equal distributions for red and green values lead to a red over green ratio close to 1, and significantly unequal distributions lead to a red over green ratio significantly different from 1. This approach is typically being applied by biologists developing microarrays, right? Now, a key purpose of this paper is to examine expression ratios. 2 points need to be taken into consideration. First, even if the red and green measurements are identically distributed, the mean of the ratio distribution will not be 1, right? Why is that? We have people from statistics here. You know. I don't know the answer to that. I'm just asking, right? Right? And the second, the hypothesis test needs to be performed on expression levels from a single microarray. Now, a salient factor in using expression ratios rather than expression differences, you want to use ratios because different genes will have different levels of response, right? So you don't want to just take the differences, right? So is that gene expression levels are determined by the intrinsic properties of each gene, which means that differences in expression levels vary wildly among genes, regardless of the truth of the null hypothesis. Therefore, it is inappropriate to pool statistics on gene expression differences across the microarray. Right? So labeling the red and the green microarray values for the genes by, at the first spot by R1, G1, second spot R2, G2, and so on, right? So the desired hypothesis now becomes that the mean of red equals the mean of green at each and every spot, right? And the alternative hypothesis is that the means are not equal, right? And the test statistic that you're going to use for testing this hypothesis is the ratio of RK and GK, the red intensity to the green intensity, right? So this requires, so if you're going to do a hypothesis testing using this ratio, that means you have to calculate the probability density function of the ratio, right, of the two random variables, right? So this requires finding a critical region for TK, recognizing that the mean of TK under the null hypothesis is not 1. Now it is well known, okay, and again, you guys that are in communications and all, it must be known to you, okay. Apparently it's well known that working with ratio distributions can be problematic, right? And recent research on the matter is generally confined to normality studies of the ratio distribution and numerical calculations. However, as we now discuss, a special situation arises for gene expression that permits a more detailed statistical analysis as well as hypothesis tests and confidence intervals based on a single microarray. The main thing that happens in this particular problem, all right, there's a biological argument that's given here why the coefficient of variation is the same. The ratio of the mean to the standard deviation, right, is going to be the same at each and every spot. Okay, that's the assumption that they're making, and they've given some biological justification for that. Okay, if you make that assumption, then what happens is when you calculate the ratio density at each and every spot, it does not depend on the number of the spot. Okay. So every spot has the same density. Okay, so it becomes like independent, identically distributed samples. Okay, and so you can pool across the entire microarray to calculate whatever parameters you need to in the density function. Okay, so that's the main idea. So there is this biological justification that's given here. You can read that. Okay, why that is correct, right? Or at least it is justified. See, many times in research, doing the work is not that difficult, right? But you might have done something, but you have to sell it, okay? So you have to find a justification. So they may have worked, right? I don't know, right? But they might have worked harder on developing the justification. Because if you make an assumption and that cannot be justified, you try to publish the paper, they'll knock your paper down in no time, right? So they have done the work to come up with this justification, right? So there is a constant coefficient of variation C for the entire gene set, right? So the standard deviation at every spot, right, is some constant multiplying the mean at that spot, right? Both in red and green channels, right? So under the null hypothesis, the red and the green have the same mean at every spot, right? So if you let mu k denote the common value, right, of the mean of both the red and the green at the kth spot, the condition of equation 3 tells you that the standard deviations are also going to be the same, right, for both the green and the red. And from the experimental protocol, we are assuming that the red and green are independent, identically distributed normal random variables. So now you're going to do some math to calculate the density of this ratio x over y, okay? And you know how to get the density function for the function of random variables, right? Basically, you first find out the distribution function, and then you differentiate them, right? So you're going to do exactly that here, right? So if x and y are continuous random variables, t is the ratio of x to y, and x and y possesses the joint probability density function fxy of x and y, then the probability distribution function for t is ft of t, right? Is the probability that the random variable capital T is less than or equal to little t, right? But random variable capital T is x over y. So you have x over y, right, must be less than or equal to little t, right? So now you'll consider two cases. Little t is positive and little t is negative. So you get these two terms here, right? And now this probability just can be written into, because it's over two variables, right? X and y. So you basically can write it in terms of these integrals, right? So here you're integrating from minus infinity to ty for x. So x is staying less than or equal to ty. And for y, you're integrating from 0 to infinity, so y greater than 0 is covered, right? Same thing over here. Okay. Now you've also made the assumption that capital X and capital Y are independent, right? So that means you can break these guys up, right? I mean, this joint density you can write as fx of x times fy of y, right? Furthermore, to get the density function, this is the distribution function, right, for the ratio. So you will take the derivative, the partial with respect to t, right? When you do that, what is going to happen, right? First of all, this ty is going to get plugged into the x location, right? And then you differentiate with t, you will get a y coming out, right? In the other case, you'll get a minus y coming out, with the result that this thing here, when you differentiate with respect to t, you get this, right? See? X got replaced by ty, and then you have a y coming out. Same way, a minus y came out from the second term, x got replaced by ty, right? And because you assume that x and y are independent random variables, right? So you can write down the joint density as the product of the marginal densities, right? So you got this. So you apply equation 5 under the normality, independence, and constant coefficient of variation condition. So now, since microradian intensity measurements are positive, densities for both red and green values are assumed to be 0 for negative arguments, right? The error created by the simultaneous normality and positive value assumptions is negligible because measurement intensities are sufficiently positive to render the portions of the left tail of the ratio distribution falling to the left of the y-axis negligible. And again, I mean, these are justifications that they have put in, I don't know how strong those justifications are, right? So now, here, you're going to now apply it to the kth spot, right? So f of tk, the ratio at the kth spot, right, will be integral 0 to infinity, right? And see, t is the ratio of green to red to green, right? So this was red to green. Which one was y and which one was x? All right. Okay. Let's see. T equal x over y. Okay. So, and here we are saying it's red over green. So x is red and y is green, right? So you can rewrite that integral, right? So instead of x and y, now it's written in terms of the green, right? And so now you're going to say that you're going to neglect this integral, right? Because you're saying that for negative values, you don't need to worry about negative values of the intensity. Again, I mean, you have to justify, okay? Obviously, they justified it enough that somebody accepted the paper, right? Right? But, I mean, why would I mean, otherwise, out of the blue, why would you drop this term? Okay, they want to get the mathematics to work, right? But then you have to justify, so they put in some justification over there, okay? So you may or may not believe it, but, the paper got accepted, right? So, no, but see there, like they didn't put in the justification just for putting in the justification, okay? They put that in to make life simpler for them and still get the paper through, right? That's why they put in the justification, right? I mean, it's not for the fun of it, I just put in terms and then take them out, right? If I get stuck, then I try all these tricks, okay? But anyway, so you'll be left with just this term, right? And you made the assumption that these are all normally distributed. So that's why you get 1 over sigma 2 pi e to the power of minus tg minus mu rk, right? From this. And then there'll be one more term, right? One more, density function for the normal distribution, right? That you'll have to put times dg. That's what you'll get in the next line, right? See? That's the other term, right? Times dg, right? So then you simplify and you can write it like this, right? And let's see, so this is equation number 6, right? This is equation number 6. Now, he tells you that the second equality follows from the positive value assumption, right? Okay, now I have to go from this equation to that 7, okay? And what I showed in the explanatory material is how to go from 6 to 7. Okay, you need to read, you need to read the paper in order to understand all the details, okay? I'm trying to give you the big picture, right, and put in some of the steps, but you actually need to sit down and read the paper, okay? Now, you might say you don't care because now it's next generation sequencing, okay? You don't want to read it. That's fine too, okay? Nobody is forcing you, It's a free country, right? So you don't want to read it, you don't read it, okay? But yeah, if I were you, I would probably read it at least once, Although it's like, 17 years old, right? This has been referenced many times. It's got like close to 1,000 citations, right? Yeah, if you go and look on Scholar Google, it's got like, so because it's the first paper, right? On the image analysis of cDNA microarrays, right? There are other groups that did the microarrays around the same time, right? There's a group at Stanford that did it. But those are like just biologists, okay? There's no combination of biology and engineering, right? So this one has got, those probably have more citations because those are biology papers with data in them, right? But this also has like close to 1,000 citations, maybe even more. So then from here you have to go to this equation, and this should look familiar, right? I have this equation in my PowerPoint slides, right? So this is the net result of making all these assumptions, right, is that you have come up with the density function at the kth spot, right, which does not depend on k. So the density function of the ratio is the same for each and every spot, right? That means we are in business. We can do maximum likelihood estimation, right, regarding the, treating the ratio at each spot, right? Like one more sample from the same distribution, right, independent sample. I can go ahead and calculate the parameter C, right, use maximum likelihood estimation, right? So they go a step further because 7 is an approximation to 6, okay? And they go ahead and calculate the approximation error also. It's not too bad, right? So now you calculated the probability density function. So now I need to calculate the parameter C, right? And so to get the parameter C, right? So typically, C needs to be estimated from the data. So using the density function from equation 7, we can obtain a maximum likelihood estimator for C. And we'll write down the likelihood function, take the derivative, right? So the likelihood function, you write it down, basically is the product of intensities like this, all right? Take the log of that, right? So you'll get your log likelihood function, right? Then take the derivative, set it equal to 0, right? And I have that done in that explanatory material, right? It's done over there. So you come up with this expression for your C hat, the estimate of C. Once you have the estimate of C, you plug it in here, right? Now you got your density function, right? You want to see what, you want the probability mass in the neighborhood of 1 to be, let's say, only 5%, right? Or something like that, okay? Then you can see, if you're outside that, you can say with 95% confidence that you're different from 1, okay? So that's the whole idea here. Okay. Building up the probability density function, right? And then using that to, come up with a confidence interval, right? So that you can say that if the intensity lies either, so if it lies below some number, right? Or let's call it -1. If it lies above some number, we'll call it +1. and then if it lies in this interval, it's going to be 0, which means that it is almost the same as 1. Okay, the ratio is almost the same as 1. So you can break it up into 3 different classes based on the ratio. Ratio is significantly higher than 1, significantly lower than 1, right? Or ratio is almost the same as 1, right? So that's the idea, and this is just providing the tools, okay, to come up with the probability density function. And then I think he has some polynomial fitting and things like that, you know. I mean, these are some extra things that they have done in this paper, right? But the important thing is this and the derivation of this one, right? The next step is that all these things, you know, we have assumed that the red and green have the same intensity, right? So that means you are working under the assumption that in each channel, right, the conditions are more or less the same, right? Now, if it so happens that one channel has got a much, you know, more intense laser beam, right, then you should not see whether everything is in the neighborhood of 1 or not, okay? You should see something else, right? I mean, so there will be a scaling factor. So that's what is done in the next section, right? So he's talking about uncalibrated signals, all right? And there he'll just use the fact that when you multiply a random variable by a number, right, how the density function scales, all right? So the null hypothesis of equal means is appropriate for calibrated signal acquisition. But in practice, this may not be the case. So we consider the uncalibrated situation in which the means of the red and green signals are related by a constant amplification factor, right? M, let's say. So the mean of R of the red signal is M times the mean of the green signal, right? So if M is greater than or equal to 1, then the red signal is stronger than the green, right? And we can follow the same derivation as in the calibrated case, except that now the ratio density has got 2 parameters, C and M, right? And this will result in the recursive relationship like this, right? This is the same form, and this is something you've done as undergraduates, okay? When you multiply a random variable by A, okay, how the density function is altered is 1 over AF of the original random variable divided by A, right? So that's what's happening here. But now, see, in this function, there'll be 2 parameters to estimate, right? M and C. Right? Earlier we just had C, okay? We assumed that the 2 channels have equal intensity. They're similar. So now you have to estimate M and C. And I think the rest of the paper, they're actually developing an algorithm to estimate both M and C, okay? So you are going to, like, so you have to now estimate both M and C. So here 1, 2, 3 up to 6, all right? This is basically an iterative procedure. Okay, you'll first update M, right? Then update C, right? Go back and forth until you converge, you know. So repeat steps 2 through 4 until a satisfactory result is obtained, right? So, and usually in about 5 iterations or so, you will converge to the correct value, right? And they used a polynomial as a function of C, right? They went ahead and calculated the mean of the ratio distribution using polynomial regression. All right, any questions? So again, I mean, as I said before, you know, I'm giving you the big picture, right? I'm giving you the roadmap. You have to go and read the paper, right, in its entirety. It's not that difficult because whatever steps were skipped, I have put those in, okay, in that handout. So you can go and read it, right? But again, I mean, if you don't care, that's different, right? So, and then using this density function, right, they came up for 95%, okay? What is the lower limit, okay? For different values of, what was it? Oh, if instead of working with the density function, you just fit a polynomial to it, okay? With parameters A1, A2, A0, A1, A2, A3, okay? Like a third order polynomial, right? You fit that polynomial and then, you know, you're saying that the density you're going to approximate with that polynomial, right? And so what is the lower limit and what's the upper limit? Like for a 95% confidence interval, the lower limit is this one, upper limit is this one, okay? And so on. And so then they measure the goodness of fit and things like that, okay? Now, what is this thing going to be used for? In this particular paper, right? They wanted to find out the genes which are differentially expressed between the 2 samples, right? In this case, it's a cell line, right? Which are the genes? So using this method, you know, they were able to, you know, find out some gene, I mean, they were able to confirm that some genes they expected would behave differently between the two cell lines, okay? That expectation was confirmed, right? And then in addition to that, they picked up other genes, right? Which people didn't know that would behave differentially between the two cell lines, right? So they picked that up. So you could get more information because, you know, and here you're, see, if you don't use this kind of signal processing, then you will just see with your naked eye, you know, which one is green, which one is red, and then you're going to try to draw a conclusion, right? But here, this is like more mathematical, right? More systematic, right? Then you got your, you did a hypothesis test, right? And then you can come up with a list of the genes that are differentially expressed. And towards the end of the paper, I think that conclusion is given. See the experimental results? See, they had the two. So consider the superimposed microarray image from the red channel and the green channel that's shown in figure 2. The full array contains 1368 clone segments, a total of 88 ratio samples, whose ratios are believed to be about 1, right? Were used, and then the result is as follows, okay? They got M as this value, C as this, and with a 99% confidence interval, if it lies between 0.566 and 1.977, you're going to say that it's almost 1, almost the same as 1, right? And based on this interval, 92 ratio samples are found to be significant, right? Significantly different from 1, okay? Which genes are differentially expressed between these two samples, right? Not at the same level in the two samples, right? They found 92 of them. Of these 70 were found to be significant using the inappropriately narrow confidence interval of reference 4. So in an earlier paper, 70 of those were picked up. So they picked up another 22 using this method here. Okay. And you can use the same kind of technique, I suppose, for, you know, doing even with next generation sequencing, right? If you want to see which in RNA-seq which genes are differentially expressed, okay? I guess the mathematics will be different, okay? There will be different assumptions, but again, you are sophisticated math people, okay? So if you can put in the right biological justification, you are in business, right? You'll write a paper and you'll get published. So. All right, any questions? So on this paper, all right, I will give you some homework to do, right? Like some data is there, some synthetic data, right? And you will, you know, go ahead and, you know, like do the hypothesis testing and, you know, figure out whether red is significantly different from green and so on. I used to give this for the undergrads, all right? Because there is an undergrad version of this class where they don't have to present a paper. They will take the tests, all right? And then they need homeworks, right? So you have homeworks on classification and so on, classification, clustering, expression prediction, and then also on the image analysis of microarrays. So it should be quite straightforward for you guys to do, but you know, it'll give you something to do, and I changed the thing a little bit. Earlier on, your presentation used to be what, 50%, right? Now I made it like 25%. So you get to do some homeworks and turn that in. You know. Although, you know, for you it's probably kind of easy, but you know, if you're not familiar with classification and all these things, it's an opportunity to learn, right? So Friday there will be a class, okay? I decided against going to that meeting, right? So there will be a class and I will get started with classification, right? And most probably today when I send you the recording of this lecture, right? That time I will send you the first homework, right? Okay. So at least you have some work to do, right? And in the meantime, keep studying the biology. Remember, there is a test that is going to be there on November 3rd, right? And everybody should be doing better on this test than they did on the first one, simply because you're getting tons of time to study, right? Right? I mean, we already finished the molecular biology last week, and then you have 3 more weeks to study. Right?
The test is just about biology?
Yes.
Just about the biology part?
Yeah.
So it's everything before classification?
Before this chapter, yeah.
Okay.
Yeah.
We don't have like a test on this?
Why, you want a test or what? No. You know, for this, what the real test will be, see, no, not even homework. You guys are going to work on some projects, right? Remember, many of you are, so you're going to apply, you know, the methods to next generation sequencing and, you know, analyze their data and all that, okay? So that will be the real test, okay? So, yeah, but in this course, it will be basically the homework, and you can, you have looked at one paper, okay? I'll discuss one more paper that is on gene expression prediction. But I think now that you know all the biology, okay, and many of you are, stochastic process whiz kids, okay, like you're wizards in all these stochastic processes. So since you know the biology, you should be able to read any paper in the literature, right, and understand what's going on, okay, because your stumbling block was the biology, okay, it's not the math, right? So you can start, looking at different papers, to figure out which one you're going to present in the class. Because as soon as I finish all these topics, all right, then we'll switch to presentation, okay? So that time I will relax like you guys and you will present, okay? So, okay, yes, sir. Yeah, go ahead.
I have a question about the content of the second test. It's going to cover everything from the beginning, or?
No, it will cover everything after translation, yeah. Yeah, that's quite a lot of materials, but go and learn it once and for all, okay? And once you know the biology, you'll find that a lot of these papers are easy to read, okay? For me, I might have trouble in the math because I'm not, a stochastic wizard, right? I might become someday, but not yet, okay? But for you guys, that are doing all 303 and all other kind of classes, so for you it should be a piece of cake, huh? Especially if they gave you a hard enough time in that class, this should be a piece of cake, Depends on who taught you, I don't know. Okay, any questions?